All right, let's get started. Since I can't see my slides very well from here, I'll just come down. I'm Dusty Majumdar, the CEO of PredxBio, and looking forward to this session. Thank you very much for coming this afternoon. It was a little ways off from the actual exhibitor center, so really appreciate taking the effort to come all the way. PredxBio has been around for 5 years now. Chakra Chanibotla, sitting over there, started the company, the founder of the company, around 5 years ago. And what we do really is focus on AI-driven spatial biomarkers. And our special sauce really is doing everything in a way that's hypothesis-free, and our AI algorithms have explainability embedded across the entire pipeline. So that's kind of been the way we have distinguished ourselves, not only getting accuracies of prediction all the way up to 90+%, but also being able to get into the why behind the predictions. Why are certain cohorts of patients responding? Why are certain cohorts of patients not responding? So that's kind of been our mantra and focus the last 5 years. Very glad to see Nature journal carry a full issue on spatial biology, calling it spatial awareness. So awareness of cells with respect to each other in the neighborhood, how they interact with each other, how they tweet to each other, and how they conspire with each other to move the disease forward. Interestingly, recently, George McNamara at Johns Hopkins pointed out that Chakra's paper in Nature Communication was the first one that brought the term spatial biology together. Now, I'm believing what he's seeing by doing his research on PubMed. And if that's true, we are really proud of the fact that we have the person that started off this field over 5 years ago, and now there's 2,500 papers out there, and some of the leaders in this field are in the room, so feel very privileged to recognize them. I'm going to have a panel in 20 minutes or so. I'm going to ask the panel members to come up here and will introduce themselves. Most of you have seen who the panel members are, and it's a very distinguished panel that we're going to get on the stage. One of the things that we do at the moment is go all the way from discovery, drug discovery, to actual deployment of the spatial biomarker in clinical practice. Now, that's not easy, because sometimes when you do drug discovery, you're starting with a 40-plex, 60-plex, 100-plex panel, and you got to get to a point where the spatial biomarker is actually deployable across different sites. So this is what we have been doing for the last 5 years. We try to get into the mechanism of action in the early stages of discovery, maybe use a panel that's 8, 10, 30, 40 biomarkers. Once we understand very clearly the predictive capability of that rather large panel, and do our analysis in a way that's completely unbiased and has explainability embedded across the AI algorithms. I forgot to mention that Chakra came from Jeff Hinton's lab. Some of you may know that Jeff Hinton is the godfather of AI, and a couple of weeks ago, Jeff Hinton won the Nobel Prize, so Chakra was happy as clams to know that from the University of Toronto. Once we figure out a complex panel, or maybe not a very complex panel, even with 6 biomarkers, we're capable of getting into the mechanism of action. We go into a clinical practice where we can actually deploy a biomarker which is fast, which is robust, which is cheap. And that's what we have been doing with a number of different pharma companies quite successfully over the last few years. Some of our collaborators are in the room. In the past, we were inspired by all the work that Akoya did on immuno-oncology, and we were focused monomaniacally on IO. Now we have shifted to ADCs and multispecifics over the last few years, and the combination of IO and antibody drug conjugates is something that's growing very rapidly out there. I was at the ADC conference in San Diego just last week, and I at least saw a few dozen papers on combo drugs between IO and ADCs. And the mechanism of action for these are very poorly understood at this point of time. So a huge opportunity there. One of the things that we do is we tackle the toughest problems that are out there in spatial biology. So, looking at cell distribution and sizes and types of cells and segmentation, those are the table stakes. We take it to the next level. where we try to predict response to therapy with 90% confidence across a range of different therapies, across a range of different biomarker panels. We are able to get from a rather complicated, complex biomarker panel in discovery, where we actually uncover the mechanism of action, all the way to a CDX panel. That's going to be a lot simpler. It could be a couple of IHCs and H&E combined, right? So that's going to be, and then, we have also worked on the diagnostic space, being able to discern diseases like ADH without ambiguity, and then combining H&E and IHCs to get virtual multiplexing. We'll talk about some of these use cases in the next 5 to 10 minutes or so. Again, we were inspired by the fact that the trials in immunotherapy were exploding back 10 years ago, 5 years ago, but the ORR, the response rate for immunotherapy, remained in the 10-20% range with the PD-L1 biomarkers that were mainly being used and are still being used out there. We know that around 30% of patients who are PD-L1 positive are recommended for Pembro or other IO treatments. This is what happens. PD-L1 positive patients, many have hyperprogression, severe adverse effects, and in some cases, when PD-L1 expression is less than 50%, the patient responds. What we have been trying to do is get away from this. Now, it's interesting, in the world of combos of ADCs and IO, I was there last week, some of these papers were presented last week, IO plus ADC gives synergistic effects in terms of the response of patients. Nobody really understands why. And guess what biomarkers are used for this? Again, the same old PD-L1, right? So these basically don't expose much about the bystander effects, the extended cytotoxicity, the reduction of immune suppression, the enhanced immune activation that happens as a result of bystander killing effects in these systems. That's what we feel is a huge opportunity for spatial biology to uncover. And again, as I said, it's kind of interesting. Even when you're combining IO and ADCs, the PDL expression, low and high, pretty much gives you the same response rate. Just take a look at the middle, right? 71 and 64% response rate. So this is what we're trying to get away from, the historical approach, where everything is jumbled up, right? To the approach on the right-hand side, where you can clearly separate the responders versus the non-responders. As a result of this, and most of you in the audience know this, stratification and selection of patients, improving the drug efficacy, developing companion diagnostic tests, all that becomes possible. And I don't think I need to lecture this audience about what's going on today in terms of failure rate of clinical trials. One of the things that we have done, though, working with pharma companies, is try to really uncover that hidden circuitry of cancer in phase 1. Not wait for phase 3 and fail, but phase 1, where we actually have a fairly complex biomarker panel that uncovers that mechanism of action, and we are able to predict with around 90% plus confidence what's going to happen in phase 2 and 3. So getting to the why earlier. In this work, we have been inspired by Leena Gandhi, who was at Dana-Farber a few years ago, where she talks about the fact that the focus is now shifting to phase 1 clinical trials. And it's really with that kind of confidence when you go into phase 3, you can actually get a faster approval. And we have seen this even with pharma companies that I was in Basel a couple of weeks ago, and Roche just came up with this paper where they're talking about patient enrichment in phase 1, early phase, and there's a lot of effort going on in this direction. And I think it's pretty obvious to this audience that if you do that, you probably have a higher possibility of getting to a high probability of approval. Any biomarker, as we know, improves the probability of approval of a clinical trial. But when you go to spatial biomarkers with this kind of accuracy, so taking a step back, why is this happening? We believe that, well, as we all know, that today's biomarkers are mainly genomic-based, genomic expression-based. Our belief in PredxBio is that genomics probably exposes around 2 to 5% of that hidden circuitry of cancer, the mechanistic underpinnings of cancer. That is what we are trying to go beyond at this point, because cancer biology is immensely complex. So this is what we have been doing, powering the next generation therapeutics, revealing the cancer biology with AI-driven spatial biomarkers so that we're able to expose the heterogeneity in the tumor microenvironment, reveal actionable biology, power clinical trials, and enrich patient populations. ultimately to get a deployable test that's simple and it's cheap and it's fast out there in the market. A few years ago, Chakra and the team had the realization that heterogeneity in the tumor microenvironment is not random. There is an organizing principle to heterogeneity. That organizing principle is revealed through the microdomains. And we'll talk about the microdomains in a second in terms of how we actually discover these in the tumor microenvironment. From the microdomains, we're able to get into the network biology of cancer, and from there, we are able to expose the various pathways. When you do that, you not only get accuracies of the order of 90% compared to the PD-L1 based tests, but you also reveal why certain patients are responding and why certain patients are not responding. That is what we have published the last few years, as you can see over here, in several marquee journals, and we continue to do that across immuno-oncology, ADCs and combos, and across not only immunofluorescence, but also lately spatial transcriptomics, IHCs, and H&Es. Again, a couple of things that are very important in terms of what we do. We're able to work with small cohorts of patients because we have explainability embedded across our AI algorithms. We don't need hundreds and thousands of patients to train our AI algorithm because with each tissue that we analyze, there's 1 million cells on a slide, and we learn everything about the cancer biology that we can. And once you do that, once you get to the biology of cancer with every single analysis of the cell, you don't need hundreds of patients to train your algorithm. The other thing that we are able to do is predict with 90% confidence. So not only the what and the how, but also the why behind why are certain patients responding here. So this is just a cartoon of what we have been doing with pharma. We typically get the images from pharma, and then the magic starts, which is looking at the complexity of the tumor microenvironment from a standpoint of the various kinds of cells here, tumor cells, immune cells, stromal cells. We're able to capture the cell-cell communication, looking at the ligand exchanges, as well as the microdomains, which are constellations of these cells coming together in a specific spatial orientation. What is very important here is this is not about composition only. This is what many other companies do. Just look at the composition and call them spatial neighborhoods or cellular neighborhoods or niches or whatever. What we do is we actually look at the spatial orientations of the cells with respect to each other, and we try to discover these microdomains which are identical in terms of spatial orientation of the various cells across the tissue. Once we're able to discover these organically in an unbiased, hypothesis-free way, we're able to correlate that to the outcomes. So that's basically what the 25 plus papers that you saw all talk about, and that's what we're going to show. So a couple of things that happen when we do that. You get true explainability from a systems perspective. You expose emergent biology, the mechanistic underpinnings, the crosstalk. You also, and we'll show this in a second, we do unbiased cell phenotyping, which is very important in terms of being able to capture the cancer biology and get down to a very simple panel in phase 3 or for CDX. We talked about the microdomain discovery and network biology. This is the SpaceIQ platform that we have developed. We can take data from proteomics, spatial transcriptomics. After some very basic quality control, this platform is fully automated. We are able to do the unbiased phenotyping using this tree and leaf recursive algorithm, which basically classifies the different phenotypes. And within a few minutes, we're able to get the tumor cell types, the fusion cell types, the macrophages, the immune cell types, et cetera. And we also know their spatial position with respect to each other. So think of this as a GPS system that we have that uncovers these microdomains on the right-hand side. Once we know the microdomains, we're able to correlate that into the network biology of cancer, and that's where the why comes in. So let me show you some use cases. Before that, a couple of quick things. We are able to actually capture, with this unbiased hypothesis-free method, the cells in transition. So we know that cells in transition, let's say from epithelial to mesenchymal transition, are very critical to capture because sometimes they correlate directly to the outcomes and the interaction with therapy. And so it's not only the red and the green, but also shades of yellow in between that we're able to capture. And Chakra over the years has developed this entire suite of tools that we use, the Bayesian network interface, spatial parametric model, point-wise mutual information. He's going to talk a little bit about that. And of course, the counterfactual analysis that we have embedded. So some use cases, then I'll get the actual scientists to come and talk about what we did here from a biological perspective. So first use case, identifying progression of colorectal cancer accurately. We're able to do that. This was published in Nature Communications and Cell Reports. We're able to do that with 93% accuracy. The interesting thing here is, there you go. We're able to do that between stages 1 and 3 using pretty much the same structure of the microdomains. So our belief in a number of studies has been that the microdomains are formed very early in the evolution of cancer. And this is really important because as immunotherapy and other advanced therapies come from the later stage to the earlier stages in cancer, it's going to be critical to understand the microdomains in the early stages. We also were able to expose the network biology of cancer, as you see here on the right-hand side, from the microdomains. On the left, you see the microdomains for patients in which the cancer recurred, and on the right-hand side, no evidence of disease after 8 years. This is for diffuse large B-cell lymphoma, so not only in solid cancers, from the tissues in the bone marrow of semi-solid cancers like lymphoma, we have been able to actually identify responders versus non-responders with 93% accuracy. The company that we worked with was over here before we came in and helped them out, and this drug was actually commercialized. This is lung cancer. We're going to hear a lot about this study with Dr. Arutha Kulasinghe sitting here in the audience and in a few minutes on the podium. This was to identify responders versus non-responders. Chakra is going to talk about this in a second with SpaceIQ platform, where we had a mean AUC of 0.88 in terms of predicting overall response and exposing the network biology of cancer as well. Similarly, for not only overall response, but overall survival, look at some of these AUC values. And again, Chakra will go through some of the science here on the network biology. Well, this is something that we'll talk a little bit in the next section of the talk that Chakra is going to give. And then how exactly do these microdomains drive response and resistance? And here what you basically see is that some of the interactions that are more frequent in responders and less frequent responders represented by the thickness of the lines and how it all comes together. So you'll have to wait for a couple of minutes before you really get into some of the science behind this. And of course, we see very different kinds of microdomains in the responders versus the non-responders. The last one is kind of interesting. This is something that we've been actively working with several companies, and I'll show you a generalized case for this, building actionable prognostic biomarkers, and then you go to the phase 3, where you're able to reduce from a significantly large panel to a much smaller panel. So 40 to 60 plex MIF panel to start with, and then reduction on the panel, further reduction here, and then really simple panel here. The beauty of this is if you do this analysis in the way we do it, in an unbiased, hypothesis-free way, using that recursive tree and leaf algorithm that you just saw, you're able to maintain the AUCs, because here you reveal the mechanism of action. So the IO60 panel would be something that we could actually use here, and then slowly, as you go along the clinical trial continuum, reduce the number of biomarkers significantly and still maintain the accuracies. If you don't do it in a hypothesis-free approach, first of all, you get pretty crappy AUCs, and then this decreases, this dramatically goes down as you reduce the number of biomarkers. So Chakra, I would call upon you to take the discussion to the next level in the next 10 minutes or so. And what I want to say is that some of us have been in this field for 30 years. Remember a time when cancer was defined by the tissue of origin. The last 20 years, more and more by tissue and mutation. We're entering a new era where disease is going to be determined more and more by multi-omic spatial biomarkers. And the kind of AI algorithms that are used and the kind of hypothesis-free analysis that's used and explainability that's embedded into the algorithm is going to be critical to have some of these answers that we are seeking today. Come and visit our booth. Some of you have already seen the Apple Vision Pro in its full glory and exposition of the tumor microenvironment. So I invite you to come and spend some time. There's a poster today, later in the day, and Filippo is going to give that. And I think we go into Chakra's presentation. Chakra?